Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go. Let's make great science together.
Okay, so let's dive right in. You know how axolotls can just regrow a whole leg if they lose one? Perfectly.
It's incredible.
You always kind of assumed the cells doing that rebuilding just had this fixed map, right? A positional memory telling them where to go.
That was the standard idea, yeah. Locked in from development.
But get this, what if that memory isn't permanent? What if it can actually be, well, rewritten during regeneration? And even stranger, it's easier to rewrite it in one direction than the other.
That's exactly the counterintuitive finding we're diving into today. Welcome back, everyone.
We're digging into some fascinating research on axolotl limb regeneration, specifically how they remember to rebuild and how flexible that memory really is.
Right, so axolotls are, the champions of regeneration. Not just skin, but complex things, limbs, jaws, even bits of brain.
It's wild. And the key thing is this structure called the blastema. After an injury, cells nearby sort of reset themselves a bit.
Exactly, they dedifferentiate and form this mass of cells, the blastema. But critically, these cells aren't blank slates. They remember where they came from.
Their positional memory.
Right.
Like a GPS coordinate for the limb.
Precisely. Shoulder versus fingertip, front versus back. And that guides the whole rebuilding process. We knew this memory existed from, old school grafting experiments.
But the how was the big question mark. What's the actual molecular code? How does an adult cell hang on to this address for ages and then just reactivate it?
Yeah, what is the memory made of? And figuring that out is huge, not just for understanding salamanders.
Right. It's fundamental biology, cell identity, development. And, the dream is regenerative medicine. Can we get human tissues to do this instead of just scarring?
That's the long-term hope. And for limb regeneration, the anterior-posterior axis front to back is super important. Signals like shh, sonic hedgehog from the back are crucial for patterning.
Okay, but how do cells know to switch on in the posterior, the backside, after an injury, based on their old position?
That was the mystery. This study aimed to find the molecules defining that AP identity, maintaining the memory, and then seeing if it could actually be changed.
All right, let's get into it. First step, figuring out where that crucial signal comes from in the regenerating limb.
You'd logically think, okay, the cells that made when the limb first developed embryonically. They just keep doing it, right?
Seems straightforward. That lineage carries the torch.
But using some really neat lineage tracing, they found something pretty surprising. They tracked those embryonic expressing cells and found that most of the cells pumping out in the regenerating blastema weren't actually descendants of those original embryonic ones.
Wait, really? So other cells were turning it on?
Yeah. Cells outside that original group were activating after the injury. It suggests the memory or the potential to activate isn't just passed down rigidly. Other cells must somehow acquire it or retain the latent ability.
Okay, so it's more dynamic than we thought. So how did they hunt for the actual molecular memory code then, if it's not just that one lineage?
They looked at the connective tissue cells in the adult limb. These are known to be major carriers of positional memory. They compared gene activity, basically what genes are switched on, between anterior and posterior cells.
Looking for the differences. The posterior signature.
Exactly. And out of hundreds of genes that were different, one really popped out. A transcription factor called HAN2. It was way more active in posterior cells.
HAN2, I've heard of that. Isn't it involved in limb development in, mice and chicks? Posteriorly.
Spot on. It's known to be expressed in the posterior developing limb and helps turn on there. So the big question was, could HAN2 be part of the adult positional memory mechanism in axolotls?
Okay, makes sense. How do they check that?
They generated some special axolotls, ones where they could track HAN2 expression directly and also trace the descendants of cells that expressed HAN2 during development.
Clever. So tracking the gene and the cells that made it.
And they found HAN2 is continuously expressed in those posterior connective tissue cells in the adult uninjured limb. And importantly, during regeneration, HAN2 expression actually increases in the blastema.
Before STIRS turns on.
Yes, before skin kicks in. And following that developmental lineage...
The cells that made HAN2 way back when.
Right. They confirmed those cells form the posterior part of the adult limb, they stick around, and crucially, those HAN2 lineage cells in the blastema are the ones that give rise to the new SHEK-expressing cells during regeneration.
Ah, okay. So HAN2 is in the right place at the right time, and its cellular descendants are the ones making shh. Looks like a strong candidate.
Very strong correlation. But, correlation isn't causation. Is HAN2 actually needed for this? Is it required for the posterior identity and the sh signal?
That's the key question. How do you test necessity?
Gene editing time. They used CRISPR-Cas9.
Ah, the molecular scissors to mess with the HAN2 gene.
Exactly. They created axolotls with mutations in HAN2. These are sometimes called crispants because the mutations are mosaic, affecting some cells but not others.
So what happened when they disrupted HAN2?
Well, first, it caused pretty severe limb development problems, missing digits, things like that. Very similar patterns to what you see if you block sh signaling itself.
Okay, so it affects development like sh does. What about regeneration?
That's the crucial part. Mutating HAN2 significantly reduced, or sometimes completely blocked, sh expression in the regenerating blastema after amputation.
Wow. So lose HAN2, lose the sh signal.
Pretty much. It's strong evidence that HAN2 is necessary for proper sh expression, which is essential for posterior patterning during regeneration.
Okay, necessary. Did they do any other tests, like functionally?
They did. They used a classic experiment called the accessory limb model, the ALM.
Right, where you trick the limb into making an extra one.
Yeah, you transplant posterior skin onto an anterior wound. The mismatch tells the cells, build a new limb here, and the posterior graft provides the necessary sh signal.
So they did that, but with skin from the HAN2 mutant axolotls.
Precisely. They took posterior skin from the HAN2 crispants and grafted it to an anterior wound.
And did it make an extra limb?
Nope. Unlike normal posterior skin, the HAN2 mutant posterior skin completely failed to induce an accessory limb.
So without HAN2, that posterior skin just couldn't signal properly. It lost its posteriorness in that sense.
Exactly. It confirms HAN2 is required for that functional posterior identity, specifically its ability to kickstart signaling. Interestingly, the cells didn't just become anterior either. They seemed to lose that signaling capacity.
Okay, so HAN2 is necessary. What about the other side of the coin? Is it sufficient? If you put HAN2 somewhere it doesn't belong, can it make that place posterior?
Good question. That was the next test. They engineered transgenic axolotls to express HAN2 basically everywhere in the limb, even in the anterior region where it's normally absent.
So forcing HAN2 expression where it shouldn't be, what happened?
The results were quite dramatic. Misexpressing HAN2 in anterior regions induced sh expression in the wrong places, ectopic expression.
And that messed things up.
Big time. It led to extra digits polydactyly, which is a classic result of too much or misplaced sh. In some more extreme cases, they actually saw entire extra limbs budding off from anterior tissue.
Whoa, so just adding HAN2 was enough to tell those anterior cells, okay, you're posterior now, start building posterior stuff.
It really seems that way. It's powerful evidence that HAN2 alone is sufficient to activate that whole posterior signaling program.
You said sometimes it messed things up completely?
Yeah, they found that if the HAN2 expression was really strong and widespread, covering the entire limb area, it could actually shut down limb outgrowth altogether.
Huh. So it's not just having HAN2, but maybe the difference between where it is and isn't.
That seems to be the key insight. The boundary or gradient of HAN2 expression looks crucial for proper patterning, not just its presence.
Okay, this is fascinating. HAN2 is necessary. It's sufficient. It's a major player in setting up that posterior identity. But now, the really mind-bending part you mentioned at the start, this idea that the memory isn't fixed.
Right. This is where the study gets really interesting and challenges some long-held assumptions. Is this positional memory set up by factors like HAN2 permanent? Or can it be changed?
How did they even test that?
With some elegant transplant experiments using fluorescent markers, they could label anterior cells one color, posterior cells another, and track their fate.
Okay, walk us through the first one. Testing if anterior memory could change.
They took labeled anterior cells from a donor axolotl and transplanted them into the posterior side of an unlabeled host limb. So you've got marked anterior cells sitting in a posterior neighborhood.
Okay, so before injury.
Before injury, the cells just sat there still looking like anterior cells, keeping their label, but then they amputated the limb, forcing regeneration through that area.
And watch those labeled anterior cells in the blastema.
And here's the kicker. As the blastema formed, those transplanted anterior cells, now surrounded by posterior signals, started expressing a posterior marker. They effectively flipped their identity.
They rewrote their positional memory.
Yeah.
From anterior to posterior.
Exactly. And it wasn't just a temporary thing. This new posterior identity stuck. They followed these cells through a whole regeneration. They contributed to the posterior parts of the new limb. Then they did a second amputation.
And the cells still acted posterior.
Yes. They had stably adopted this new posterior fate. The memory was rewritten, and it held.
That's kind of wild. So the memory can be changed. What about the other way around? Can you turn posterior cells into anterior ones?
They tested that too. Transplanted labeled posterior cells into the anterior side of a host limb, amputated, and watched.
Did they flip?
Mostly no. The posterior cells largely kept their posterior identity. They didn't switch to anterior. In fact, sometimes they mess things up by creating little ectopic posterior structures in the anterior part of the regenerated limb.
Okay, so there's a definite bias. It's easier to go from anterior to posterior than the other way around. Memory is stable normally, but gets plastic during regeneration, and it prefers that A to P switch.
Precisely. And they backed this up by looking at the overall gene expression. The transplanted anterior cells really did adopt a full posterior transcriptional signature after the switch.
Right.
It wasn't just one marker changing.
So what's driving that switch? What's making those anterior cells flip when they're put in a posterior environment during regeneration?
The prime suspect, given everything else, had to be shush signaling, right? It's the dominant signal coming from the posterior side.
Makes sense. So did they test it?
They did. 2 key experiments. First, they repeated the anterior to posterior transplant.
The one where the cells flipped.
Right. But this time, they treated the animals with a drug that blocks the shush pathway during regeneration.
So if shush is the signal telling them to flip, blocking it should stop the flip.
And that's exactly what they saw. Blocking signaling prevented those transplanted anterior cells from acquiring the posterior identity. They stayed anterior.
Okay, so shh from the surrounding posterior tissue is necessary to drive that reprogramming.
Definitely seems that way. Then the second experiment addressed sufficiency, is just adding shush signal enough to make anterior cells flip, even without being physically next to posterior tissue.
How'd they test that?
They took anterior blastema cells already in that plastic regenerative state, and treated them with a molecule called SAG, which artificially activates the SH pathway.
So just bathing them in the flip signal.
Essentially, yes. And they found that activating SH transiently during that regeneration window was indeed sufficient to make those anterior cells stably acquire posterior memory.
Wow. So they started expressing hand too.
They started expressing hand too, and amazingly, they even became capable of producing SH themselves in a subsequent regeneration. It confirms SH can reprogram A to P, but crucially, it seems to work best during that plastic phase of regeneration.
Okay, let's try and pull this all together. What's the big picture emerging from this work?
Well, it really highlights this molecular circuit, this feedback loop between hand 2 and SH as being central to posterior identity in the axolotl limb. Hand 2 primes the posterior cells. They make SHIK after injury, and that SH signal can then reinforce hand 2 and posterior identity, even reprogramming nearby anterior cells.
And the major innovation seems to be showing that this memory isn't static, it's dynamic, especially during regeneration.
Exactly. For really the first time, we have a molecular handle on how this positional memory is encoded in adult tissue, but also how it can be actively changed. It enters this plastic state during regeneration, receptive to signals like SH, particularly for that A to P transition.
What does this mean for other research? Where does it lead?
For basic biology, it's huge. It gives us a concrete mechanism for memory and plasticity. It helps explain things like posterior dominance that researchers had observed for years. Why is A to P easier? Now we have a potential molecular reason. And it begs the question, what controls the switch between stable memory and this plastic state? Understanding that is key.
And for the applied side? regenerative medicine.
It's inspiring. Could mammals have analogous systems, maybe involving different molecules like Hox genes, which are also involved in spatial memory? If we understand how axolotls make their cells plastic and receptive to reprogramming cues like shh, could we potentially find ways to induce a similar temporary plasticity window in human cells after injury?
Instead of just scarring, maybe promoting actual patterned regrowth.
That's the ultimate goal. It really shifts the focus, doesn't it? Maybe the secret isn't just finding stem cells, but figuring out how to make the cells already there flexible enough to rebuild properly, making the map adaptable.
Yeah, it makes you wonder. If mature cells have stable memory, but blastema cells become plastic, maybe finding and controlling that transition point, the moment cells become receptive to new positional information, is the real key to unlocking complex regeneration in animals like us, who usually just, well, patch things up with a scar.
That's a really provocative thought. Is that the bottleneck we need to overcome, manipulating the state of cellular memory itself?